These majestic, enigmatic creatures are actually essential wardens of our marine environment. Found in every ocean across the world, they play a fundamental role in ensuring balance and stability in our oceans. Sadly, this role is jeopardized by overfishing and habitat loss. Around 3 billion people rely on fish as their primary source of protein. Ongoing declines in shark populations could have devastating and unpredictable impacts on marine food webs, and even our own food supply. The plight of sharks and rays was the spark for the Save Our Seas Foundation, founded over 15 years ago. Through the funding and support of research, conservation, and education initiatives, we are dedicated to safeguarding the future of sharks and rays. Today we have 3 centers based in Seychelles, South Africa, and the USA. Since 2003, we have funded over 300 projects on over 60 species in over 50 countries. We have also partnered long-term with several projects, including the Shark Spotters in South Africa, the Shark Lab in the Bahamas, and the Manta Trust. We're passionate about our mission: protect the future of sharks and rays and our oceans for generations to come. We can and will make a difference. The Save Our Seas Foundation.
Well, hello, everyone. Welcome to tonight's Save Our Seas Foundation Distinguished Speaker Series. My name is Lance. I'm coming to you live from the Museum of Discovery and Science in Fort Lauderdale, Florida. Tonight, we're going to be bringing you another researcher having to do with all kinds of really cool research, having to do with a new or a species of skate tonight that lives in Tasmania, kind of a elusive species. So welcome. We're going to have a great time. Welcome back to those of you that have come back again and again. We're happy to see you. Here at the Museum of Discovery and Science, I am the Director of Environmental Sustainability. We do a lot of really interesting things here at the museum. If you're ever in the area, please come by to visit. Here's our museum. We're, like I said, Fort Lauderdale. Our mission is to connect people to inspiring science. That's what we do every day here. And we do this in 4 different areas: physical science education, early childhood education, health and wellness education. But my specialty and one of our 4 core pillars is environmental sustainability. So we're going to be bringing you a little bit of that tonight, connecting you with some really amazing research that's happening in the shark world, skate world, the ray world, and having a good time. Again, thanks for the Save Our Seas Foundation, of course, for making this possible. We really appreciate their support in supporting the whole series. You can find the whole series actually on the Museum of Discovery and Science YouTube channel, where you can find 3 years of the speaker series going back for a while now, all interviews with researchers. So take a look, take a minute to like and subscribe there, and you'll be able to find this video there relatively soon too. Welcome, Dr. Awruch. How are you tonight?
Hello, how are you, everyone? Hope everyone listened to me okay. There is just a truck outside my window.
No, we can hear you just fine. It's great. It's great. Dr. Cynthia Awruch is joining us tonight to talk about her research in Tasmania about an elusive species of skates. She'll be telling us all about her research there. Dr. Awruch's passion for marine life began in her childhood during fishing trips with her grandfather. Born in Buenos Aires, Argentina, Dr. Awruch completed her undergraduate and honors studies there before moving to Australia in 2002 to pursue a PhD at the University of Tasmania. Her primary research focuses on aquatic vertebrates, so that's animals with backbones, especially fish and chondrichthyans, so cartilaginous fish, using physiology and reproductive endocrinology. So this is hormones, reproductive hormones in these animals, as tools for conserving and sustainably managing marine wildlife. She also conducts fundamental research in these areas as well on several different species. Currently, Dr. Awruch is a lecturer at the Institute for Marine and Antarctic Studies at the University of Tasmania, where she also serves as the research ethics manager. Additionally, she holds a senior research role at Argentina's Center for the Study of Marine Systems under the National Scientific and Technical Research Council there. Over the past 2 decades, her research has spanned both Argentina and Australia, focusing on non-lethal methods for shark and ray conservation and the impact of marine pollution on cartilaginous fish, on their health, their reproductive systems. As a founding member of the Oceanic Chondrichthyans Society, Dr. Awruch was instrumental in organizing the Society's first conference in 2007. Her work aims to expand biological knowledge while developing non-destructive research techniques, contributing to more effective conservation and management of marine species. So it sounds like you're doing a lot of really cool stuff. We're happy to have you here tonight, Dr. Awruch.
Hi, Silan. Hi, thank you. Thank you everyone for being there, and hello, and yeah. Let's get started. And once I start share the screen, I may not be able to see everyone, but yeah, I'm more than happy to be interrupt anytime during the talk to answer any questions and make the talk very dynamic. And if you think that I put a very scientific term that you don't understand, please interrupt me, and then I try to explain everything, but just in case sometimes we forget, yeah, sure. Interrupt me, I'm happy to answer.
Yeah. Thank you for that invitation. If anyone, go ahead and you can go ahead and pull up your screen if you'd like. And while you're doing that, if anyone does have any questions for Dr. Awruch tonight, please put them in the chat. I will see them and we'll ask them on your behalf. You know, if it's a burning question right then and there, I might be able to sneak it in during the presentation, but we will also have a Q&A session at the end. So don't be shy. Feel free to ask questions as we go through. All right. Dr. Awruch, I see your slides. Everything looks great.
Good. Okay, today we are going to talk a little bit about this species. It's called the Maugean skate in Tasmania. I just want also to acknowledge the other 2 people very involved in the project, which is Dr. David Moreno and Dr. Jason Simmons. And let's get started with the story and hope you enjoy it. I want to introduce the species. You may know already what are constrictions and what I'm talking about, but just in case, I'm always a nerd that I want to put the species where the species belongs. And as you know, constrictions are shark, rays, and skate, and chimeras. And the sharks and rays and skate, there are a group called Elasmobranchi, and because I like to understand where words are coming from. You could see that Elasmobranchi means that they have this branchia, which is, you know, beside and it's very flat. What holocephalids are those fish, those kind of constrictions, they are more bulky in their head. And it's very interesting because I do work a lot with the brain of the animals to understand reproduction as well. And taking the brain of these animals is very difficult because the head and the upper jaw are very strongly linked. That means it's very important to understand we have this group of constrictions, but the Maugean skate is part of the Elasmobranchi group, and they are within an order called Rajiformes, with these flat fish. And all these skates, all the skates are part of this group. You know, they are the skate in general, very robust animals, and they don't move as beautifully as the rays. And where these skate live, and this is very interesting. I'm trying, but it's not, there it is. The skate was originally recorded in around 1988, when they start to have some encounter of this new sort of species. But it wasn't really until 2007 this first paper was published talking about the skate particular and finding the place for this skate in the world. The skate live in I will say two areas, but then when you realize that actually maybe only one area in Tasmania. You know, you have a map of Australia here, just in case. Here is Tasmania, is this beautiful island under, but it's part of Australia. And there are these two harbors, the Macquarie Harbour here and the Bathurst. And I'm going to concentrate on the Macquarie Harbour. And this area is very particular because it's a very small area. It's a very complex habitat for any species. The water, if you go there fishing, the water looks very brownish, and not because it's brownish because there is rubbish there. It's just because it's a tannin-sustained surface, because all the trees around, the type of trees, it's an area with very, very low oxygen. It's a very poor area in terms of nutrients. I mean, the species is found there, only there. And it's very interesting because the species, the cousin of the species, they live in South America, in Chile, in Argentina. That means it's very interesting because it's this very old animal that become very separated to Australia when Gondwana, I don't know if all of you know or remember when we talk about Gondwana, and we'll have all this land together and they start to separate millions of years ago in South America and South Africa and Tasmania and all the area of New Zealand. Means for some reason the skate got there to Australia and stay there. One very similar species we have in South America, which is very interesting because that helps us understand how the world used to be in the past. The species is oviparous. They lay eggs, which means that the embryos develop outside the mom. And the other thing that is very interesting about the harbor is very shallow. It's not a deep water area, but because the condition of the harbors, the animals behave like deep water species with low light. And this is very interesting because a lot of the behavior that we see around the species there are really more related that we see with deep water species, but this is a very shallow area. And before we start talking about the skate, I want to talk to you about the history of the harbor, because that will explain what is happening with the skate these days. It means around 1800, the first settlements start, there was the logging industry, and then they built a massive seawall in the harbor, and the seawall is made by rocks. And the reason of the seawall is at the end of the harbor in the channel, when the harbor is linked to the sea, they build this seawall to try to, for this channel to become still smaller and not get very big. I mean, there is all these rocks there, a lot of construction happened there. Then in 1881, they start mining. And this is a very important thing, you know. Mining is always an issue, and particularly at those times, there were no regulations, a lot of water from this mining going into the Macquarie Harbour. Then in 1974, they dam in one part also of the Gordon River. It's one of the rivers in this Macquarie Harbour, in one of the branches of the harbour, to build an hydroelectric power generator. And this also is a big of a problem. In 2000, they developed escapee salmon fishery. And in 2009, the expansion of the salmon aquaculture. I mean, all these components are very important to understand what happened in the harbor and what is the situation in the harbor at the moment. And then, having that in mind, why we decided to study these species? And the reason is, around when I was doing my PhD, around 2002, I have a colleague at the time, a friend. She was also doing a PhD on skates. And during her PhD, she was studying all this skate in Tasmania. And I remember she was saying, we hardly see any of the Maugean skate. We don't understand what happened. Then people start thinking about it. And so, okay, maybe it's time to study what happened with these species. And one of the things you do when you start thinking about the species is you try to understand how this species is based in this world, is sitting. Is this species endangered? Is threatened? Can we fish it non-stop? And this is when all these listings come around. I don't know if you are aware of those listings, but Australia has listings and the world has listings. And the Threatened Species Scientific Committee assessment, this is an Australia committee in 2004. This is a long, long time ago, decide that you have different criteria to decide if the species is endangered. And even criteria 2 and 3 are met because the species live in a very small area and there are very low number of individuals. You have to start thinking that less than 2,500 is one of the criteria to classify the species. But we don't know much about what happened with the population in the last 10 years or 3 generations. What is the population size of mature individuals? And what is the probability of extinction in the wild? That means we need to understand more of that. Of course, all this information is about to be reviewed because we have new information, and this has been a long time ago. And also, Australia has what we call the EPBC Act, that is the Environment Protection and Biodiversity Conservation Act, and they did declare the species as endangered. Tasmania also declared the species as endangered, and the IUCN Red List, the International Union for Conservation of Nature, also did it. Means Tasmania, at the moment, has a recovery team thinking about what is happening with the species. It's not a legal plan, because for that we need legislation, but we have a team that they meet very often and put on the table everything that is needed for this species to be conserved. And when I talk about what is needed, is all the people or all the industries that may influence the survival of the species in the future. The thing that we don't have yet is what Australia called the Threat Abatement Plan, the TAP. That is a plan that Australia organized on threatened species. When this was treated long time ago, they didn't think that the skate has all the criteria to be in one of these plans, but of course this is going to be reviewed. With all this in mind, we start to thinking, okay, what are the problems that the species are encountered? And one is the fisheries. There is not a commercial fishery on the harbor. There is recreational fisheries that has stopped at the moment. We don't know if there is any introduced predators like seals. Sea lice, I don't know if you have encountered sea lice or if you have ever been going fishing, but sometimes sharks or any constrictian, when you catch them, have a lot of sea lice, and that is not good because can kill them. Because the harbor is environmentally impacted by the mining, by the salmon farming, but hydroelectric in the past, we need to understand what happened with the price of the species of the skate in the harbor. We don't know. There is historic pollution, and I mark this in particular because this is something that we are going to concentrate on the talk. And also, there are some environmental issues in the harbor. One of the biggest issues is climate change. You know, Tasmania is a very important hotspot for climate change. This is not good, but the harbor is even more. You know, there is very increasing temperature, low dissolved oxygen, which means it's not good for the species because it has to change areas all the time, and it's not good. That means all this warming of the environment, the low dissolved oxygen, and other things that we can control at the moment, but we need to find a way to modify for the species to survive.
Dr. Awruch, can I ask, so based on the harbors, all these impacts you're saying that are going into the harbor, right? Are there other species besides the Maugean skate that are threatened or having similar issues? Or is the
Yeah.
Maugean skate more of like an indicator species, a species like the species that if it's doing good, the rest of the environment's doing good. If it's not doing good, then that's a good indication that the environment is going downhill.
I'm going to talk about that during the talk because there are other species and some of it's perfect. And I cannot see any of you at the moment. That means just interrupt me anytime what you want because I cannot see the comments. But yeah, you will see in the talk that there are other species there and are much of these other species that we are using to understand what happened with the skate. Okay. What we know so far from the skate, this is from many years of study. We know that the species has very low genetic diversity, and we come from this idea that genetic diversity is important for a species survival. But this species doesn't have a big genetic diversity. And we don't know what. We don't know if because there was a massive reduction in population size and then the species lost the genetic diversity, or on the contrary, the species was driven from a very small pool of genetic diversity, and this is how they survive. And more studies are showing at the moment that this concept of important genetic diversity is not true for all species, that actually some species are driven by having a low genetic pool. And this is very important. Sorry if you hear any noise. This is my dog asking me for...
It's okay. We welcome fluffy friends. It's okay.
Good. But what is important is the species has a very low genetic diversity. And this is a very important information for us because I will tell later we have a captive breeding program, but we have one mom there laying a lot of eggs, and the genetic diversity of these embryos now is very similar. And we cannot put all of them back into the water because if not, we are putting in this very already small genetic diversity pool, a very even smaller genetic diversity pool, And it's very interesting because what we are finding is even the low genetic diversity is there. The species, they are like siblings, they don't get together. They don't mate together. It means there is something happening in the environment that even there are not many specimens, still they don't copulate between each other when they are siblings. Another thing that we know now is the species seems like it doesn't live for long. We need to do more studies, but there is an indication that they don't live for long. They mature to be reproductive when they are 5 or 6 years old. That means they have a very short time of reproductive space. The gestation length of these eggs when they come embryos is around 31 weeks. We don't know when they are laying eggs in the wild. We never found a pregnant females in many years of sampling, but in captivity, they are going bananas. They are laying eggs all the time. And we think because in captivity, they are sharing the best perfect conditions they can be. A lot of food availability, constant temperature, and they are as happy as they can be. And the other important thing is we did find eggs, not in the female itself, but yes, in the water, and they were around 2.5 to 33 meters. That means we don't have any evidence that they prefer one side or the other for laying eggs, which usually is important information because when we do management protective measurements, it's important to know, do we have to protect one area more than the other? Now,
what is the environment like where they are laying their eggs? Is it flat like bottom, muddy sort of like body? Is it
Yeah.
It is. Okay. So it's not like a kelp forest or something like that. It's just like flat, soft bottom mud sort of situation.
Yeah. And it's very, it's just very dark. Yeah.
What do they attach their egg cases to? Do they find some hard space to attach it, or is it they just free flow through?
Many times they are free flow. You will see sometimes the eggs are attached, and I'm going to show a egg case in a minute. And sometimes when you see egg cases that they have like what we call tendrils, that is this like long tendrils, you know that these eggs is going to be attached somewhere and it's going to be flowing with the water. Sometimes they just like eggs there, you know. They may be between some more, you know, planty areas, but still they are not going to move as much.
It's interesting. Okay.
The other thing we know is they feed with small crustaceans, which is important also to understand because they don't eat whatever they want. They just, I mean, they don't eat everything. They have a very specialized diet. That means we also have to be protective of all these animals because this is what they feed. And then there was a study done about how they move. And what they found is they usually move between the 5 to 15 meters in the water column. They don't move much. They can do it, but they prefer a small home range. And what was very interesting and sad in some way is we always thought the species both in Bathurst Harbour and Macquarie Harbour. And as Macquarie Harbour is a very contaminated area, Bathurst Harbour is very pristine. And that was really good because one of the ideas was to say, let's start all these captive studies. And then when we have a lot of embryos, we can put them back in the harbor, in Bathurst Harbour, because the species is there. And one of the things of rewilding, of putting species back in some habitat is because the species has to have been there before. But they did a massive, massive study doing environmental DNA. For everyone that may not be aware of that it is like when you go to the water and you collect water, and in the water, because there are tissue and skin from the animal, then there is this DNA that is coming from that lives just floating there in the water. That means you can get a good amount of water and then amplify the signal of the water in terms of this DNA and see if there is DNA of this species. And they did a very, very, very intensive study from many locations in the harbor in Bathurst, and there is not sign of the species. The species was there in the past, very few animals, but it's not there now, which is very sad because now we can only concentrate in Macquarie Harbour at the moment.
Wait, was there a history of them being in the other harbor?
There is a history of, I think, 4 animals that were recorded, but that's it, you know. And who knows, maybe in long time with the species was separating from each other. Some went to Macquarie Harbour, some got in this separation in Bathurst, and they maybe the number was so small that they just disappeared. We don't know, but it's not there now. And then what happened is, and how we start in our study is around 2013, 2014, they start to, because most of a lot of the information I show is from many, many years of study up to now, including what I'm going to like go back in time. And then in 2013, 14, when they start doing the studies, one of the things is we need to understand reproduction. Means Dr. Jeremy Lyle, that he used to lead this project in the past, now he retired. He contacted me because, as Lance said, I work on non-lethal ways to understand reproduction. And he contacted me and said, Cynthia, can we use your technique and use hormone to understand? And this is how I came on board in this project. I mean, I'm not going to concentrate much of the talk on that, but just for you to understand in the past, to understand when the animal reproduce. And when the animal reproduce is crucial information that we need to use for conservation, because it's very important to understand when the animals mature and start producing babies. We need to understand where, because then when we do conservation, we say, okay, this area, you know what, don't touch it because the animals are reproducing. Or this size of animals, don't touch it because these animals are sexually mature and are ready to reproduce. I mean, reproduction is a massive, massive information that is crucial for any management plan that we do. I mean, in the past, we used to go and we kill the animal, we open and we see the gonads. And you can see an ovary here of the female. And what you can see here is the female has all these follicles, and each of these follicles is going to be ovulated and will become an embryo. Of probably not all of them, because not all of them are going to be fit, but this is how it works. It means what we start to do is, instead to open the female and see all that, we start taking blood and then measuring reproductive hormones, like we have testosterone, estradiol, progesterone, and then validate what the hormones are telling us with the ovaries and also with the testes in males. And finally, we develop a technique that's okay. Now we know that what we see in the hormones translate to what happened in the ovary. We can use the hormone information to understand reproduction. And the first time we started working on that was in 2007. That was part of my PhD. And it wasn't actually until 2014 that we finally published a paper where we didn't kill one animal, none.
Oh my gosh.
We all were able to understand reproduction just using hormones. That means with that information in place, then we did this. We catch a lot of animals, we took blood, and also what we did is we did ultrasound, like we do in our ovaries in females, in women. And then you can see here, these follicles, this ovary is not from the skate, it's from another animal, from long time ago from the fishermen, that I got the specimen from another animal, but it's similar. You see the follicle here, you see the follicle in the ultrasound. And what that this information is very important, because the size of the follicle is going to give us an indication if the female is ready to ovulate or the female is inactive, is not producing follicles at that time. It means the combination of hormones and ultrasound is really good. At the moment, we are working in many other techniques to understand reproduction, even from the brain without killing the animal. But this is for another talk. Anyway,
that's really amazing that you were able to do that without or killing any of the animals to study them. That's really nice.
that is really nice. I had to say, if this is my legacy to science, I can go in peace. And you can see me there taking a blood from the skate. We don't even do it like that anymore. I don't have a video how we do it now, but now we get the skate, because the skate reduced in numbers so much. At that time, we did a trip, and maybe in a week fishing, we will get 20, 30 skates, and then of course we release them back into the water. In the last few years, maybe we do a week trip with very intensive fishing, and if we get 5, it's a lot. And then, we get the skate, we put it in water, we are working in a different, but at that time, usually when we get an animal, it's very important that you have something wet to cover the eyes and to cover the body, and we take blood, we put them back. Okay, that means with all this in mind, then we start taking blood. We understood reproduction, and then in parallel when we were doing this, David came on board and said, I'm going to do my PhD. David got a scholarship to do a PhD at the university, working on the population of the dogfish in Macquarie Harbour. And then the dogfish, I call them like lab rats for sharks, because they are very easy animals to work. All the people working in physiology and endocrinology in the 1970s, in the 1960s, use the dogfish as a model because it's very easy to catch. It's everywhere. All the countries have a type of dogfish. And then when he came on board, we start thinking, okay, we start realize that we weren't getting many of the skates anymore. David came with his project, and then he said, maybe I should take blood to understand the reproductive cycle, also using hormones in the harbor. And then what we start to find is that these species that we understand very well how they reproduce in Australia, in other areas in Tasmania, around the world, in the harbor, we're doing something completely different. And that was a worry. And the worry came because what we start finding is the species, one of the things important is a viviparous species. That means that the embryos develop inside the mom. But what we start finding is this, that the size of maturity of the animals was smaller than in any other places, sometimes this is an indication of stress and pressure to mature earlier, to reproduce earlier, because they need, and you can see that sometimes in fisheries, when they, even with teleos fish, when there is a very strong fisheries, we saw that in Argentina a lot with the very important commercial fisheries for the hake, that they start to realize the fish is mature earlier to reproduce because they don't have enough, they are so stressed to reproduce, to produce, babies, to keep going, that they start doing earlier and earlier. The other thing is we didn't find a clear seasonal pattern of the species. And one of the things we found, and I'm going to explain this in these slides, is most of the pregnant females, the one that we found, and there weren't many, they were pregnant only in the first stages. Like if you see this photo here, I don't know if you have ever seen something like that, but this is an ovary of a shark. And then these follicles, each of them will come an embryo. The time of ovulation will release this area and will come through here and will travel all here. Here, the follicle is fertilized by the sperm of the male, and this is the uterus. And this is where the follicle is going to become an egg and develop and become an embryo. If you see this photo, you can see this is an oviparous species, the egg case and the embryo inside before it's going to be released. In a viviparous species, what you expect to see is all these little embryos here. And what we start to see is, we can see the egg. We knew that the follicle and the sperm got together, but we never saw the embryo develop, which is very rare in this species. I mean, it's like, okay, something is going on. One of the other things that they found is these animals, when they look at the stomach content, most of the stomach content of the animals were pellets from the salmon industry. The pellet that they use to feed the salmon, not for any other thing. That was a worry. And then we start thinking, okay, is this pellet enough energy for reproduction? What has the pellet that maybe may affect the species? And is the pollution of the harbor affecting the species? What happened with the males? In reproduction, we hardly look at the males. And in terms of I'm going to speed up a little bit because I can see the time. In terms of pollution, one of the things we start to realize, for you, if you don't know, in the last year, there are more and more work done on pollution in the marine environment and in sharks. Any shark constricting has to bioaccumulate a lot of pollutants because they are high in the trophic level, because they have a very slow metabolism, very lipid-rich livers. That means they do it. And what happened when they start to get this contaminant? Many of them are what they call EDCs, endocrine disruptor factors. And what they do, this factor, is compete with the hormones in our body and in some way affect our reproductive system and other systems. Some of the EDCs that you know a lot is the pill for females to stop pregnancy. They have a lot of estradiol, estrogens, massive EDCs, some pharmaceutical products, personal care products. I mean, heavy metals, anything, and it doesn't matter if you have a short or a long-term contact with those, can affect your reproductive system. That means you can see that, we live in this area close to the industry where we are, a lot of pesticides, a lot of sewage discharge, all this is affecting the animals. And this is how it works, These chemicals will mimic the hormones and we compete, in general, in our body for the hormones to work. We have a receptor of the hormone, then the hormone comes, they get together, and they say to the body, do something. When this signal is not done by our hormone, by another chemical similar to the hormone, it's a problem, because then the effect is not happening. Or the other thing that happens is blocking this receptor, and also is not happening, or can break down the system. This is a big, big problem. And it's something I am really passionate about. That means we come with this idea. Let's use the shark and the Maugean skate get some analysis and try to understand and see if the pollution is really affecting these species together. Because the Maugean skate, because it has been declining, we have to be very careful, and sometimes we need some dead animals to be able to see the liver and see other part of the body. And we can get that from the fisheries, but there is not fisheries on the Maugean skate. That means what we are doing at the moment, we are measuring reproductive hormones in both species. We are measuring ketone bodies. Ketone bodies are molecules that are produced by the liver when we don't have enough energy from glucose. That means when we are diet or food deprived, we will start using ketone bodies. That means we want to see if this species using ketone bodies. If that is the case, they are not getting enough energy from other sources. We are measuring different contaminants. We are measuring vitellogenin. Vitellogenin is a protein that produces the follicles that they are filled. Males shouldn't have it. That means if we start to see that there are males with the vitellogenin, we have an issue with high levels of vitellogenin, because then it's okay, they may be producing estradiol, which is more a female thing. It shouldn't be much of a male thing. It's a big information of disruption of the endocrine system. And in invertebrates, we can see that a lot. We start to look at the sperm quality in males, see histology of the dogfish. Sometimes you can see in the histology, we cut the ovary and we put it under the microscope and we see the tiny cells. Sometimes there is evidence of health diseases. And we are doing all this in the Macquarie Harbour. And then with the dogfish, we are going also to a very pristine area and we are going to compare the results. And our project also has a lot of educational program. What we found so far is high level of contaminants in the Maugean skate, in the harbour, comparative to even other species around Tasmania. And we have high level of other endocrine disruptor in the dogfish in Macquarie Harbour. And we are about to go fishing in summer in Australia, which is your winter, to get dogfish from pristine areas. Okay. We are still doing everything. I don't have result for everything. This is what I could show at the moment. But then we are going through. That means maybe in next year I can come back and tell you more about it. And another thing, we have an educational program, and I can show you this. How is this word? Oh, okay. What is that? Here, I show you very quickly. And then this is very beautiful. We have kids that they talk about the skate and they try to save the skate, and it's very beautiful. That means we try to educate the kids because we think it's very important for the new generations. And then we are doing this associated work that has to do with, we have animals in captivity. We are using these animals to ensure a population in captivity, to also have the embryos to understand how to work with these embryos in terms of climate change and pollution. A new methodology to understand if we are really capturing all the animals, because at the moment the species has been declining, but now it's not declining in the last 2 surveys. That means we need to understand, did we have any bias in our sampling? We have to be very cautious with that. And then, and keep monitoring the population, because it has been really bad for so many years that we don't really understand at the moment. Has stopped, but has an increase. And I just want to say thank you to Save Our Seas, because this is possible because Save Our Seas, and to Jason and David, my partners in crime.
That sounds like a really amazing project. And I really love how you put like all these pieces together, right, to come out and solve the problem, because the problem is that you don't have enough specimens of the skate to study, but through another species that's relatively plentiful, you can draw some conclusions. And that's a really interesting and unique way, I think, to come to solve that issue. So yeah, I'd love to hear more. I mean, that'd be really great to have you back. We're getting a few questions rolling in.
You need to understand that.
Please go ahead. No.
No, I was just going to say that as the manager of the animal ethics at the university, I'm very strongly about animal welfare.
Yes.
That means you need to really justify, using an animal for anything. And every time we catch a skate, it's like 15 minutes of stress to be sure we handle everything properly, the water is properly, because anything that would happen to any animal will be terrible. Yeah. That means we need to find these surrogate species to understand more. Yeah.
Well, it's so interesting to also think about assessing reproductive health of a skate, approaching it this kind of the same way you approach it with a human, right? Like we do blood tests, we do ultrasounds, like that's how we assess the health, reproductive health of humans. So why can't we just apply that to other animals as well? We're animals just like they are. So, and oftentimes some of the same hormones can, are part of it, right? So it's great. And we're big fans for animal welfare here. So kudos to you for doing that. Because it can be a little more difficult, right? You have to be a little more creative, but I think you get good results when you do that, right? We have a few questions coming in about skates in general. We'll start there as a category, I guess. Someone was asking, how fast do they move? Are they quick swimmers? Do they like to just hang out on the bottom?
No, they just hang out in the bottom. They are not quick swimmers. Yeah.
What are they looking for to eat? They're just like stuff hiding in the mud, worms.
Yeah, little crustaceans. Yeah, mainly. Yeah, that they are hiding there. Yeah.
Okay.
And if you see the feet and the mouth is very like back in the body. It's not on the front like sharks. Or rays, it's just at the back. That means they are swimming like this, and then they find, with the nose, they try to move the sand and find the prey.
Oh, so they use their front to kind of like shovel the sand, mud and stuff. Okay, that's cool. How do you catch them whenever you go out sampling? How do you go about doing that?
We catch them on nets. That means we put the net for maybe 40 minutes and we catch them. And this is the reason we try different sizes of nets. And then the only time we try to avoid the net is an area where there are a lot of dogfish, because then you get too many dogfish in the net. It's not good. But now they are trying this new technology. Hopefully it's like this. It's like the harbor become like an ultrasound machine where you can see if the skate is there or not. Yeah.
Really?
Because, we try to do, they try, this is not what I do in the project because I'm the physiologist in terms of reproduction, but doing a survey, like VRU, as they do in other sharks, I put praise and they see if the shark, but the water is so dark that it's very difficult to see. That means they have to come with more creative ways to see what animals are there and how many. Yeah. Right.
That's interesting. So someone was asking, just if you could just go over really quick, the difference between a skate and a ray, because they do look very similar, but you were saying like maybe one difference is like the skate has a mouth that's further back, but what are some other differences between skates and rays?
Usually the group of the rays, the tail, can you see, is very long. The swimming is more like flying.
For a ray.
For a ray, the skates are this is more flat, and also they are not pelagic. They are really living on the bottom. Okay. The rays become more pelagic many times, but this is like in a very general. Yeah.
Cool. So a few other questions.
And I'm sure if you talk to a taxonomist, it will give you 10 more time, more. But
that's okay. It's okay. I mean, they do look kind of different. Usually skates have more of a pointed snout on them. So more, like you said, their tail, their caudal fins are a little bit different.
And very flat.
Do they have a stinger like a ray would? A barb?
No, they don't have a stinger, luckily, because I've been stung by the ray in the past, and it's not a nice feeling. No.
Oh, you've been barbed by a ray? Oh no.
I've been barbed,
yeah. Well, I'm glad that that's not a risk when you're studying skates.
Yeah.
And just a few other little questions. that came up. Now you were saying that there is a similar species to the Maugean skate, but it's in Argentina, you were saying, right?
In Chile, yeah.
In Chile,
yeah.
On the Pacific side, right? So,
and in Argentina, but Chile as well, yeah.
Yeah, so here in Florida, there's a lot of endangered species. One of which is the Florida panther, and to assess to help alleviate the issue of low genetic diversity, right? They've introduced another species that can hybridize with the panther. So they bring in mountain lions, and they've shown that they will reproduce and make a hybrid, but similar enough offspring. And so and help to enhance the genetic biodiversity of panthers. Could something like that be done with the Maugean skate? Could we bring in a similar species from Chile and see how they do in the lab, and and then I don't know.
I don't know, because I think one of the things that it's important is we start to recognize that maybe the low genetic diversity is important for this species. Okay. And the other thing is, when we have them when we start having them in captivity, it was very surprising, because we it may take forever for the species to acclimatize and start reproducing. Right. But the female just went bananas and never stop. Actually, the problem that we are having now is she's laying too many eggs. And then we cannot put all these embryo bags in the water, because we'll affect this very low genetic diversity, because we are putting like 25% of one pool of gene. It doesn't work like that very well. Yeah. I mean, it's not a possibility. We have other alternatives in terms of things that we could do in terms of, but the captive breeding program is working really well. We just need to be ensured that we bring more males to create a bigger pool. But it could happen, yeah. I mean, I'm sure there are a lot of also legislations in the rewilding of when you put these species back into the water, and a lot of considerations, but yeah. But we have in Australia species in the wild that we always thought that some of the species live in the north and some more in the south, but in the middle we have a hybrid population by choice. We didn't do it; they did it on their own.
Right. Oh.
Some of them. So let's play with some friends here. Yeah.
Oh wow, that's cool.
I think what is fascinating, Lance, is we're still learning. I think animals keep surprising us of what they do in nature. You know,
right?
It's not black and white. Right. Definitely black and white. Yeah.
What was that? Life always finds a way, right?
Exactly. Yeah.
So, well, thanks for answering that. But so we have time for a few more questions. Just in, I know you're doing your research now. You're doing a lot of the data gathering, but you know, if you had the opportunity to do more, you know, if you were given a blank check and said, Dr. Awruch, go do what you will, what would you do? How would you expand upon this? What are those questions that you still want answers to that would help you know, fill in the gap of missing information?
I think more than missing information, I think we are covered a lot, and I will go more deeper into the disruption of the reproductive system. I think we are touching the surface. There is hardly anything done on sharks, because it's very difficult to study constrictions in those things, because you know, they keep them in captivity and it's not easy models. But I will get more money to fully understand how this works. We have some project with other species more in Argentina when we do study the connection between the brain and the gonads. And I would like to translate this into Australia to understand that. And also I would like to have a blank check to clean the harbor, you know, to have all this. There is an oxygen program at the moment trying to understand if we can clean the harbor, because it's such a gorgeous area. And yeah, that will be also a dream come true.
Right. Yeah. Yes, of course. Well, how can people that are interested in following you, how can they learn more about what you're doing? How can they stay in touch or just get involved in ocean conservation in general? What would you say?
Well, sorry, I forgot to put my email there, but you can Google me and you will find my email or David's email. And of course, you are more than welcome to contact us. Go to the Save Our Seas. We put blogs as much as possible. And then there is also contact information that you can contact us. And for me, in terms of helping is, I think just be conscious of what's happening. It's good enough, and see how you can help on that, and see, you know, sometimes you can contribute and start doing a research project, and sometimes you just contribute to stop polluting the environment and be aware of climate change and whatever we do. You know, we always say each contribution of each of us is not enough, but when we pull of us together, and sometimes it's pressure to our governments, it's pressure to the aquaculture farms to do the right thing. I'm not saying to close them, nothing like that. We have to come to live all together, particularly in this world where there is less food. We just need to be sure that we pressure enough for the people to take the measurements to do it properly. And pressure our governments, I think, is the biggest power we have as a community. Yeah.
Right.
Yeah.
Yeah. Yeah. Well, that's great advice. Being more aware of your surroundings, your environments, and educating yourself on issues, right, that you may not know a lot about, but that are really important. Well, Dr. Awruch, thank you very, very much for coming tonight and speaking with us all about your research. We really, really appreciate it. And like you said, I'd love to have you back when you have more results. That'd be fantastic.
Yeah. Thank you very much. And sorry I didn't put my email, but I can send it to you, Lance, later, and then you can.
Yeah, of course. Well, if anyone's interested in learning more, you can. The Save Our Seas Foundation website is a great place, to saveourseas.com, to learn all about different researchers and what's happening, about Dr. Awruch's projects, and specifically, you have a blog, so a few blog posts about some of your research on the website as well, and they can read more about you there and follow along. Join us next time for our last speaker of this year's series on December 5th, 6 p.m. to 7 p.m. Eastern. We're going to be speaking with Brian Keller from Saving the Blue, all about protecting silky sharks here off the coast of Florida and in the Caribbean. So we'd love to have you there. Said that's going to be our last one for this series. So you can sign up at mods.org/STEM Learning Series, or you can scan that QR code right now and sign up. And of course, follow us on all the socials. YouTube channel, you can find a Mods YouTube channel. You can find all of our videos going back, like I said, 3 years of Save Our Seas Foundation Distinguished Speaker Series. Check them all out. It's a good watch. Thank you again to the Save Our Seas Foundation. We really appreciate it for making tonight possible. Thank you again, Dr. Awruch, for being here and sharing your awesome research and your knowledge and experience. And thank you to all of you for joining. We'll see you next time.
See you next time. Thank you.
All right. Bye-bye.